This is my presentation for my biochemistry YouTube project. First, to introduce myself, even though I'm sure you already know, but my name is Ruth Black and I am a biochemistry major at Simpson College. I will hopefully be graduating next year in May of 2021. My fingers are crossed. A fun fact about me is that I am actually left-handed. Alright, enough about me. Let's get into the presentation. Today, I will be discussing FRET technology for visualizing the localization of proteins within cells. So, what exactly is FRET technology? FRET technology stands for fluorescence resonance energy transfer. During FRET, the energy from an excited molecule or donor molecule passes directly to another molecule that is nearby, which excites the nearby molecule, or also called the acceptor molecule. This is done by attaching fluorophores to the donor and acceptor molecules. FRET is used to measure the distance between proteins in cells or molecules within cells. More specifically, FRET is great for researchers to be able to determine whether proteins are interacting rather than just co-localizing with each other. However, FRET is really only useful in measuring small changes in distance between the donor and acceptor molecules. An analogy that I read on microscopyu.com made an analogy comparing co-localization and interaction and the difference between the two. So imagine co-localization would be like understanding that two students are present in a large lecture hall, but not really knowing much more than that. And then interaction would be more like knowing exactly where those two students are sitting in the large lecture hall and whether or not they are sitting adjacent to each other or talking to each other. And then the optimal distance between the donor and acceptor molecules is less than 10 nanometers apart, and the ideal length is about 8 to 10 nanometers apart. When the donor and acceptor molecules are too far apart, the FRET signal that can be measured is lost. FRET is a qualitative measurement, not a quantitative measurement, and it measures the distance between proteins when the acceptor emits a wavelength. Next, we will talk about what FRET can be used for in biological systems. FRET can be used in a variety of ways. It can be used to detect mutations within DNA by using oligonucleotide FRET probes that are specifically designed to detect mutations within the DNA. It can also be used to detect how proteins are folded because since FRET is able to measure the interaction between proteins, it can subsequently give researchers more information about how those proteins are organized. FRET technology also can measure cell signaling, which involves the interaction of proteins, and so cell signaling can be analyzed using FRET technology. And finally, pharmaceutical research is a growing field that has recently been heavily utilizing FRET to learn more about medicine and how it interacts with our bodies. I'm sure there's many of other real-life scenarios and uses of FRET, but these are just a couple that I had found. So, as you can see, FRET technology has many uses in biological systems. An article that I read discusses the uses of FRET technology specifically in nanomedicines or the pharmaceutical field. So, the article that I read explains that since FRET is useful in seeing the interactions on a nanoscale and seeing how proteins specifically can interact It can also be used to see how nanomedicines are able to interact with the biological environment. The article discusses the many uses of FRET in nanomedicines. Um, One that I found very interesting was using FRET to analyze the structural features of nanomedicines. So, specifically, like how drug designers can design drugs, and they can use FRET to do that. And what will happen to the nanomedicine when it's released into the body. So, using FRET, scientists are able to see how nanomedicines interact with different tissues in the body. They can use different fluorophore pairs to assess what happens when drugs are loaded and released into the body. So the figure that I chose here shows that enhanced FRET efficiency readings on a graph would represent the successful loading of a drug. And then in the second picture on the right, reduced FRET efficiency then represents the release of the drug into the body. So when a drug is loaded, molecules are very close together, which results in an enhanced FRET efficiency. Since FRET is kind of measuring the distance between those molecules, and since they're very close, you'll get a higher reading. And so then again, like when the drug is released, the drug molecules will disperse throughout the body and result in a reduced FRET efficiency since the molecules are becoming farther and farther apart and the FRET reading is harder. And so this can help scientists answer whether or not a drug is effective in reaching the target tissues. Another figure from the same article also demonstrates how FRET works with nanomedicines. Nanomedicines and proteins can form FRET pairs, and the protein can be labeled or marked with fluorescence and then binds with the nanomedicine. If the protein has a high affinity for the nanomedicine, the FRET efficiency is increased. A high FRET efficiency would indicate to scientists that the protein is a good target protein for the particular drug that they are researching or trying to develop. Finding a good target protein is crucial in order to find target receptors on the cell membrane that drugs can target in order to be effective within the body. We will discuss some pros and cons to using FRET technology. So, before FRET was discovered, there were other fluorescence microscopy techniques, and they were kind of the only way for scientists to see the co-localization of proteins or other organelles and structures within cells. So, FRET, on the other hand, allows scientists to actually see how those proteins are interacting with each other compared to other fluorescence techniques that can only tell whether proteins within cells are associated with each other. When using FRET, energy from the donor molecule is able to be directly passed to the acceptor molecule without having to emit a photon, which makes FRET a lot more efficient than previous techniques. However, because FRET is able to determine the distance between proteins and analyze how those proteins are interacting with each other, the proteins have to be very close, like I said earlier, kind of within that 8 to 10 nanometer range, in order for FRET to be useful enough for researchers. So if proteins are far apart or if the proteins are too large, FRET actually does not work properly. So to summarize things, FRET, or fluorescence resonance energy transfer, is a technique that utilizes fluorescence to measure the distance between molecules that are close together, and it can be used in a variety of ways to see how molecules and proteins interact with one another. In the future, I think that there will be more ways to use this technology, or like this, and see maybe they'll develop it in a way that you can kind of see larger molecules on a larger scale or increase that distance between it to make it more useful in general situations. Before I end this presentation, I just wanted to include a little reflection about this project. I know it wasn't a requirement in the guidelines, but I wanted to add my own twist on the project. So, overall, I had a lot of fun putting together this presentation, and I feel like I learned a lot, not only about how to use new technology on my computer, such as these recording equipment, and integrate myself more into the online learning environment, but I also feel like I learned how to put together a professional presentation and how to research a topic that I have actually never heard of before. I think these are important skills that we don't always learn in the classroom setting, but I think that they are crucial to life outside of college and also life in graduate school. So I'm glad that I got this practice. I hope that this project stays in the curriculum for the following semesters, so that other students can have the same experience that I got. I love that I was able to make connections between emerging technologies like these and the pharmaceutical field. I'm very interested in the pharmaceutical field and how drug development works, and someday I hope to become a pharmacist. So, this technique that I learned about was very applicable to my life and also to furthering and making connections in my understanding of topics that we learned and covered in this class throughout the semester. Finally, here is a list of the sources I used to compile this presentation. Thanks so much for listening.